Today I will present you two parts of my work these last years, from synthesis to extraction and purification of valuable carotenoids, with a specific focus on fucoxantina, but we will not talk only about fucoxantina. This work is in collaboration with these people that you see on this slide, and with many companies and university academics that I had the pleasure to work with these past years. So, can you see the second slide? It goes well? Okay, perfect. So just a few words about who I am. I am currently a postdoc researcher at the Microalgae and Processes platform at the CEA in the south of France. There is a lot of sun here. I really like this place, by the way. You should come to visit. Before that, I was a high school teacher in biochemistry in Paris, also in France. I was also an entrepreneur for a few months in biomimicry. It's kind of mimicking in nature to do some innovations. It's really, I was passionate about that. We can talk about that later if you want, also in Paris. And my formation, so I have a PhD in marine biology that I completed in France. I ended it in 2022. And I have a biological engineering degree and also a Master of Transformation and Valorization of Natural Resources. These two diplomas at the same university in France, University of Technology of Compiègne. Okay. So for this presentation, I will start with a short introduction on brown algae, carotenoids, and then a specific focus on fucoxantina. I don't know at all what you know or don't know about pigments, algae, etc. So if it's too simple for you, sorry in advance. And if you have questions, don't hesitate. And then we will move to two case studies. The first case study is on the microalgae Thysochrysis lutea that I worked on during my PhD. We will talk about scaling up in 300 liters, and then we will talk about the specific fucoxantina harvesting and purification. The second case study is on another brown algae, which is Phaeodactylum tricornutum, that I'm currently working with. We will talk about medium optimization a bit and fucoxantina enhancement, and then also scale up in a much lower volume, 30 liters, and also a medium recycling process. Okay, so let's start with the introduction. So as you know, microalgae are a group of unicellular photosynthetic organisms. They are eukaryote, and they live in an aquatic environment. At the beginning, there were two big families, the green algae and the red algae. Among the green algae, you have the chlorophytes, the euglenas, also the dinoflagellates that are also red algae for some species. And among the red algae, you can find the cryptophytes, haptophytes, heterotons, and diatoms. These are some examples. And among these red algae species, we have our third big family of golden brown algae. That is what I am interested in my work. So these, not only the brown algae, but microalgae in general, as you know, they are at the base of the trophic network for primary production, and they participate in the carbon cycle on Earth through photosynthesis. And this is what is of interest here. We are going to talk a bit about light harvesting because the pigments, these valuable molecules, are at the base of the photosynthesis. And in the golden brown algae, the major carotenoid, the major pigment, except chlorophyll A, is the fucoxantina. So about the carotenoids in general on the market and on the research and development companies. So there are several carotenoids which are valuable. You have the phytoin, which is the very first carotenoid, the fucoxanthin, the neoxanthin, astaxanthin, zeaxanthin, lutein, and beta-carotene. Currently, the fucoxanthin and astaxanthin are the most promising on the market from microalgae. And on the right of the slide, you have all the, not in detail, but the biological, biosynthetical pathway of the carotenoids. So all algae don't possess all of these pathways, because the green algae, the red algae, and the brown algae all have different pigments. Finally, the pigments define the big families of algae: red, green, and brown. Okay, so within the microalgal cell, the carotenoids are, of course, used for the light harvesting. They are the first step of the photosynthesis chain that transfer the energy to between each other, the fucoxantin to the chlorophyll A, etc., and it creates the electron chain. They are very useful for membrane stabilization within the cell, within the thylakoids, within the chloroplast. They are useful for energy dissipation through photosynthesis, or when there is an excess of light. They are, of course, active antioxidants, which is also the case for human. We will see that after. This is a common property between microalgae and us. And of course, they are huge scavengers of reactive oxygen species, the famous ROS. For us, for human health, the carotenoids lower the risk of inflammation, of heart disease, of cancer. They improve the eye health and other organ health as well. They have the role of protectors in many types of cells, including neurons, which is really of interest for Alzheimer, the types of neurodegenerative diseases. And they lower the risk of type 2 diabetes as well. There are several studies about that. And so specifically now about the fucoxantin. So this molecule is quite common. It looks like every carotenoid that we know, but it has a special bond. You see in orange, and this special bond makes it quite different and very special compared to the other. So the fucoxantin is a bioactive molecule because it is highly valuable, and it is produced by all the brown algae, macroalgae, and also microalgae. Microalgae produce way more fucoxantin than brown algae. Within the body, the fucoxantin turns into fucoxantinol, which turns into amarocyxantin A, and this is this molecule, the amarocyxantin A, which has finally all the properties that we cited before, and I will detail a bit now. So the fucoxantin is known to be antioxidant, of course, because it is a carotenoid, but anti-cancer. There are a lot of studies, and I receive studies in my mailbox every 2 days on anti-cancer proof of fucoxantin efficiency, etc., etc. Fucoxantin is also anti-inflammatory, anti-obesity, anti-diabetes, antimicrobial, anti-Alzheimer, and has several protective effects. The little stars that I put with anti-diabetes and anti-Alzheimer, it's because they are not that much. I should have put one in at anti-obesity as well. I forgot. There are not that much studies on that for now, and sometimes people think it's just speculation or marketing, but we will see in the coming years. So the fucoxantin is highly valuable for pharmaceutical products, nutraceuticals, and cosmetic products. So we have already several products on the market, and we'll see that a bit after. I won't detail them now, but if you have more questions, I'll be happy to discuss that. Currently, the production of fucoxantin is natural production only. There is no chemical synthesis yet. It is only dependent on the microalgae, because we don't perfectly know the biosynthetical pathway of the fucoxantin. Everyone is looking for it because it is I mean, you can make a lot of money if you discover how to synthetically produce fucoxantin. However, this year, a study has really unlocked our knowledge on the fucoxantin biosynthesis, because a key gene of the carotenoid pathway has shown to enhance the fucoxanthin accumulation. This gene is the violaxanthin deepoxidase-like 1, VDL1. And there were already a few studies, including one of mine when I was in a PhD. I noticed that on transcriptomics analysis, we already hypothesized that this gene could have an impact on the fucoxanthin production or accumulation. The term is different. We can talk about this later as well. So if you're interested in this very nice study, I'll put the reference here. I will, I think I can send you the slides, but not all the slides. We will see that after. Some are currently in preparation for an article, so we will discuss that after. This one is okay. So just a few words about the fucoxanthin current market. So currently the commercial sources of fucoxanthin are only brown seaweeds for food, for cosmetics or for medicine purposes. It's different, but for now it's just brown seaweeds. But microalgae are the most promising because, as I said, they produce 10 times, 20 times more fucoxanthin than macroalgae, and they are often easier to cultivate. The fucoxanthin is mostly sold in Asia as a seaweed in Asia and also in the United States as a seaweed powder or edible oil, and the annual growth rate is plus 3% in average, and it's growing. And you can see by the $1 million numbers that we have here that this is a growing market. I don't have the number of 2023, sorry, but I am sure it is bigger. It is sure. I can see that with many companies I talk to. So yeah, don't worry, it will increase even more. So just to finish this slide, a few words on what species are used in what companies. So mainly brown macroalgae for food. You have Laminaria sargassum, which is a big problem in the Caribbean Sea. So a lot of potential for valorization of the sargassum species. Fucus species, Undaria pinnatifida, Ischia fusiformis. And the companies who sold the seaweed powdered fucoxanthin concentrated seaweed powder are mainly in Asia and can be sold in Asia and in Europe. In Europe, these macroalgae species are authorized, which is not the case with the microalgae species. In France, for example, we have this famous company Microfit. You can see just in the middle of the slide. Microfit, they have an ingredient from Pheodactylium tricornutum. It's called BrainFit. Actually, they have several ingredients, and they cannot sell it in Europe for now. So they sell it in the United States in waiting for their their product to be authorized in Europe. It's been years and years. They're trying to make it, to have it authorized, but not yet. It's very, very long time. The novel food regulation in Europe is wow. And the Food and Drug Administration in the United States, I don't know why they are more, they authorize more molecules. Maybe they are more advanced. I don't know, in the tests. I don't really know that. Anyway, in terms of species, currently we have two species famous for fucoxanthin production, Pheodactylium tricornutum and Isochrysis species, which is a cousin of Isochrysis lutea that I'm going to talk about after. In cosmetics, cosmetics is a very promising market for fucoxanthin because it is the fucoxanthin is anti-aging, anti-wrinkle, anti-UV, etc. And you know, it's very famous on the market. And we have such a lot of products, and we want more and more of natural product, natural resources in our daily routine. So I think it's going to be one of the biggest markets for fucoxanthin in the coming years. And in the medicine market, pure pharmaceutical market, we are not there yet, clearly, because to authorize the fucoxanthin, you have to authorize the entire species where it comes from, or you have to have an ultra pure molecule, which is very difficult to obtain. Okay, now just before to go move with the case study, just a little recap on reminder of on fucoxanthin production, extraction, and purification. So it is important for fucoxanthin production to consider all the upstream processes from lab scale to industrial cultures, because to optimize your culture medium, to optimize your light accessibility, etc., you need to start at a very small scale. And most important, you absolutely need to find a balance between the light which is needed for growth and the not too much light which is needed for the synthesis of fucoxanthin. Because the fucoxanthin is a photosynthetic pigment, the less you have light, the more there is fucoxanthin, because the cell uh, will want to accumulate a lot of pigments to capture every photon available, even if there is only a few. So it is a bit paradoxal, but the less you have light, the more the cells will produce fucoxanthin. But if you don't have enough cells, your productivity will be super low. So it really is a balance. And there is this super interesting study by Feng Zhengao et al., process optimization of fucoxanthin production with Tisocrisis lutea. It is very interesting because it compares different types of photobioreactors, of culture types, and you don't have the same. There is there is not one specific light intensity for the fucoxanthin production. It all depends on your material that you have in your lab or company. Now, a few words about the downstream processes for the fucoxanthin extraction. So it is a liposoluble molecule, so it needs to be extracted in an organic solvent. Several are present in the literature. There are a lot of literature on fucoxanthin extraction. You have the methyl tetrahydrofuran, acetone, ethanol, methanol, edible oils, ionic liquids, and the natural deep eutectic solvents. We are trying to respect more and more of green chemistry principles these last year and even more in the coming years. So we try to use solvents more environmental friendly. So we tend to forget a bit about the methyl tetrahydrofuran, the acetone, the methanol, and we are more focusing on ethanol, oils, ionic liquids, and the NADES, natural deep eutectic solvents. Just a few studies on the NADES for now. It is quite innovative for the fucoxanthin specifically. I don't know about other molecules. Maybe it's common for other types of molecules, but for fucoxanthin, no. And to respect these green chemistry principle, you have to be aware to take care of the choice of the solvent, but also on the consumption of solvent. If you use 200 liters of ethanol or, I don't know, another ionic liquid to extract 100 milligrams of dry biomass, it's not worth it. So it's always a balance here, and you have to take it into account. And for some purposes, such as pharmaceutical purposes, you might need to want to purify the fucoxanthin. Several techniques as well. The supercritical CO2, very famous in the literature. The HPLC, high performance liquid chromatography. Although the HPLC, there is a lot of solvent consumption in the HPLC. We need to use high volumes of solvent to fractionate just a bit of biomass, a bit of pigments. Not a biomass, because we have already an extract before we purify it. But anyway, HPLC is good for analysis, in my opinion, but not for purification at an industrial scale or even semi-industrial scale. And we have this very, in my opinion, very innovative technique. Maybe you know of it. It is a centrifugal partition chromatography. And this is what I will talk about later. It's part of my PhD work, and I think it really can be the future for the purification of the fucoxanthin and other molecules. It is already used for other plant molecules, quite new for pigments though. Okay, so here as well, it is important to focus on eco-purification with the same needs than the eco-extraction. So there is a difference between extraction and purification. I hope you understood that, and we will see that in the first case study. So same focus on the choice of solvents and consumption of solvents for the purification. So first case study on Thysochrysis lutea. So part of my PhD work, actually last part of my PhD work. So this algae, I don't know if you know. So it is a microalgae. It looks like a little cherry on the screen because it is with the epifluorescence microscope. It was taken in 2020 by a colleague. So what is this algae and why it is interesting? So it is an aptophyte microalga, a brown microalga, from 3 to 5 micrometers, so quite small, with 2 flagella, so it can swim. You don't need to shake them all the time. The culture, just you put some bubbling and it's okay. They don't sediment. It is really practical. Also, they have no coccoliths, so it is easier to break to look for a metabolite specific. You don't need to use beads or several processes to break it. You just put a bit of solvent, a bit of ultrasound, and it breaks. Really, really easy. And we won't talk about genes in this presentation, but this algae has a fully sequenced genome, which is really practical when we are looking for some specific genes of the photosynthesis, for example, of the fucoxantina enhancement. And above all, Thysochrysis lutea is among the best fucoxantina producers. And also, I'm not sure the best, but it produces also omega-3, docosahexaenoic acid, DHA, and SDA, stearidonic acid. I think stearidonic acid is quite original for microalgae. This is why it's of interest as well. So this microalgae is already widely used in aquaculture for bivalve, etc., and feed, and is very promising in human nutrition and has a lot of properties for pharmaceutic and cosmetic, above all because there is a lot of fucoxantina inside. So what we did to produce first the fucoxantina before to extract and purify it. So we scaled up some cultures of Thysochrysis lutea with the common strain CCAP 92714. We went from 350 milliliters to 300 liters with the same nutrients, the same incident light on the wall of the photobioreactor, continuous light, no dark light cycle. The same inoculum, I think it was 1 million cells per milliliter for each at the beginning, but according to the machines that we had, the bubbling was variable and the CO2 supply as well. But still, we maintained the same pH normally, so normally it's fine, even if it's variable. And what was super nice for us, so there are a lot of details in this experiment, but we won't talk about that, just the fact that the fucoxantina concentration, we managed to increase it by 6.5, sorry. And it's I'm not talking in milligrams per liter, because it is dependent on the concentration of algae, this unit. I'm talking in milligrams per gram of dry biomass, so independent from the volume of the algae. So we managed to increase the production by the cells. So once we produced our biomass in these 300 liters SCOBA lights, we obtained 100 grams of wet biomass. It's not much for 300 liters, but we stopped the culture really soon for analysis purposes, but we could have gone way more, way higher. So once we have our wet biomass, we lyophilized it. We obtained 30 grams. We obtained 30 grams of dry biomass, and then we extracted it either with acetone at first or ethanol for green chemistry purposes. You remember, and we will see that after. and with ultrasound. Not much of ultrasound, just like 30 minutes, it's okay to break the cells. I think some laboratories don't even bother to use ultrasounds. You just put them in the freezer and you take them back and the cell breaks. It's really easy. So at first we used 200 grams of solvent. We think in grams for this extraction, not in volume. It's important. So 200 grams of solvent for 1 gram of biomass. It's, in my opinion, quite a lot. It's not viable at industrial scale. And that's why we tried to optimize it after. And then we centrifuged everything to get rid of all the membranes and all the things we don't want. We just wanted the pigments and also the lipids because they are co-extracted as they are liposoluble molecules. So we had our extract and we evaporated the solvent to obtain the crude extract composed of pigments and lipids. In the end, from 1 gram of biomass, we obtained 300 milligrams of pigments and lipids. It was a sort of paste, really hard to recover from the balloon at this moment of the procedure of the method. So first, we wanted to know what was, before to extract anything, before to purify anything, we wanted to know what was in our crude extract. So we analyzed all the extract, the entire extract by HPLC, high performance liquid chromatography, to see the exact content. And then, very interesting point, the purification. We went to the purification of the fucoxantina. So we diluted our extract, our crude extract, our paste, into the CPC solvent system. I will explain what it is just after. And then we injected it into the CPC column. So CPC, it's for centrifugal partition chromatography. And let's talk a bit about that now. So this chromatography techniques, it's a chromatography. So it is used to separate molecules of interest. Here it's for pigments. And the CPC is based on the difference in the partitioning of molecules between two immiscible liquids. Sometimes it's solid, sometimes it's immiscible liquids. Here it's immiscible liquids. We have a stationary phase that we load at the beginning of the process, and we have a mobile phase. Altogether, it is a solvent system. And the CPC column is composed of hundreds of twin cells, as you can see on the image. So you load your stationary phase first into the twin cells, and you start the centrifugation. So the half part of half bottom of the cells are full with stationary phase. And then you let circulate your mobile phase with your extract, as we're going to see just after. So these twin cells, they are organized on the discs. There are dozens on the one discs, and several discs are arranged all together to make the column. So basically, you take your diluted extract, and with the mobile phase, you make it circulate all along the hundreds of the twin cells and all along the process with the centrifugation and with the circulation, because there is a pump. The molecules we have an affinity for either the stationary phase or the mobile phase. They are immiscible. So in the end, we will elute some fractions of the mobile phase only or stationary phase. And normally, we will manage to purify some molecules because they have more or less affinity for these solvents. Very important, this little reference. It is way too small for the importance of this reference. But Gonçalves de Oliveira Jr. et al., they published in 2020 an article on the updated pigment composition of Tizocrasis lutea and the purification of fucoxantina using CPC. At that time, it was the second paper ever on the purification of fucoxantina by the centrifugal partition chromatography. Themselves, they were based on a previous study on the fucoxantina purification with the CPC, but otherwise, it was one of the first time that the CPC was ever used to extract to purify the fucoxanthin. And in my PhD work, so we based our methodology, everything, on this specific study of 2020 to purify the fucoxanthin. So it was our study was the third of for the fucoxanthin purification, and I'm sure I can bet on it that in 10 years, 15 years, we will have a lot more interest into this technology, because it is very low solvent consuming, and you will see a lot of publications about that for sure. So let's get back to our experiment specifically. So we injected our diluted extract within the CPC column, and then we collected the fraction. We collected each 5 milliliters, but we can collect every milliliter if you want. The problem is that after you have to analyze everything. So if you have 500 fractions to analyze, it will be hard for you. So we collected 5 milliliters fraction, and we analyzed them in 2 steps. One step, but I want to talk about it today by HPLC, high performance thin layer chromatography, to analyze the lipids in these fractions, and by high performance liquid chromatography to see if we manage to separate or not the fucoxanthin. So this is a photo that I like. I did a little photo shooting of my fractions. It took me half an hour to have this photo. Anyway, these are all the pigments that you can find in a brown algae. You have all the chlorophylls, chlorophyll A, chlorophyll C1, chlorophyll C2. I cannot tell you which is which. And I'm sure the first one on the left is chlorophyll A, because there's a lot of chlorophyll A in these samples. And after you have the fucoxanthin in brown and orange, it is fucoxanthin mixed with chlorophyll C when it's brown, and when it's orange, it's pure fucoxanthin. I mean, more or less pure. We will see that after. And after you have the other carotenoids, diadinoxanthin, diatoxanthin, vialoxanthin, not astaxanthin, anteraxanthin, etc., etc. So you have all of this in like 1 milligram of biomass. I think it was worth the time of the photo shooting, because it's so impressive. So we based our method on this study that I told you about just a few minutes ago, Gonçalves de Oliveira Junior et al., published in Alga Research. So at first we did exactly, or almost exactly, as they did themselves. So we started with an acetone extract, and the acetone extract, it was not like the study. It was just for we used that in our lab, and we were used to use that in our lab, so we didn't change our methodology for the moment, because we were used to extract pigments in an exhaustive way with acetone. So we kept the acetone extract for the moment. But everything after is the same as than the study. The stationary phase was cyclohexane, not very environmental friendly for now. And the mobile phase was composed of methanol and water. Overall, the proportions of the 3 solvents, water is a solvent, were 6-6-1. And the CPC parameters, it was a small column, 240 milliliters. It's a lab scale column, but you have up to 5 liters scale column, and this is an industrial one. And so this is what we obtained. So just to explain how to look at these graphics, the first one is the percentage of each pigments present in each of the fractions. The fractions are on the x-axis. The fucoxanthin is in orange, the chlorophyll C is in blue, and the other ones, we won't talk much about them, so not important to mention. And the second graphic, it's the content in pigment in milligram per gram of dry biomass in each fraction. And the second really represents the elution profiles of the pigments. You can see whether it is well retained in the phase, or not that much. Anyway, I won't go into detail here. So what we obtained for this first test, we obtained a maximum purity of fucoxanthine of 80% approximately, 79. I precise that it's an HPLC purity because you still have lipids on these fractions. We didn't separate yet the lipids. It was not the purpose. And in Thysochrysis lutea, the lipids are omega-3, highly valuable for the brain. So it was also nice to keep them for an eventual final product. So we obtained this purity for three fractions among 50 fractions. And we had a very good recovery yield of 90% compared to the crude extract, because you remember we analyzed the crude extract at the first, and we have a partition coefficient of 0.1. So it is a bit small, this partition coefficient. And we can see on the second graph specifically that we extracted the fucoxanthine at the exact same time as the chlorophyll C. So really not good for us. And the chlorophyll C is not much in terms of milligram per gram of dry weight, but we clearly don't want the chlorophyll C anymore in our fractions. So as this chromatography was not selective, we tried first to improve the selectivity of all the process. So we get back with our acetone extract and we changed the mobile phase. Also to answer more to the principles of green chemistry, we replaced methanol by ethanol. They have not the same polarity. They don't have the same polarity, so it might clearly change the elution profile of the pigments. And this is what we obtained. We obtained the maximum purity higher, up to 87% in 3 fractions, but a recovery yield really low, 66%, not good at all compared to the crude extract. The partition coefficient was better. The previous one was 0.1. Here it's 0.2. And we can see on the second graph that the fucoxanthine is eluted at the same time as the chlorophyll C, but still a bit after. So what happened here is that we increased a bit the polarity of our mobile phase. And by increasing the polarity of the mobile phase, the chlorophyll C was eluted a bit earlier than the fucoxanthine. So this is what we focused on for the next test. We tried to increase more the polarity of the mobile phase. So we added more water in the system. So the new proportions were 2:1 for cyclohexane, ethanol, and water. So by adding water, clearly you increase the polarity. And this is what we obtained, way much better. So we obtained almost 100% purity in fucoxanthine. I said almost because all depends on the resolution of your machine. With the HPLC that we had, we could see really traces of different pigments. So I think when we are really close to 100%, we can trust the analysis, but it's my opinion. We obtained 9 fractions and not just the 3 that we had of almost pure fucoxanthine with a very satisfying recovery yield of 95% compared with the crude extract. And here the partition coefficient is much better. It was 0.5 compared with the 0.2 that we had before. And we can see on the second graph specifically that the chlorophyll C in blue was extracted really earlier compared with the fucoxanthine. And we can see the 2 peaks. These are chlorophyll C1 and chlorophyll C2. We even managed to see these peaks, which is quite rare for this. I mean, it's the only time that we managed to separate the chlorophyll C1 and C2, actually. So, very good for this solvent system. So, we validated it for now. I mean, we could have improved it, but you know what it is. It was the end of my PhD. I didn't have much time. I could have worked on it. I mean, I don't know. Another PhD would have been good to improve the solvent system, especially to replace the cyclohexane that we don't want because it is super toxic. But anyway, we focused on the mobile phase for this study, and a lot of improvements are yet to come. So we validated the solvent system. And for the last test, we tried to replace the extraction solvent, which was acetone at first, as we always did in this laboratory where I was, and we replaced it by ethanol. Ethanol is more environmental friendly, it can be biosourced, and is biodegradable and not toxic. I mean, not for this purpose, of course. So this is what we obtained, a maximum purity of 100% of fucoxanthine in 9 fractions with a very satisfying recovery yield as well, 92% compared to the crude extract, with a partition coefficient a bit lower than previously, but it's not a problem at all here because we have only the fucoxanthine in these fractions. We have 0.4 of Kd, which is fine. And you can see on the graph that clearly the chlorophyll C was extracted before, was eluted, sorry, before the fucoxanthine, and that there is no trace at all of all other pigments in these 9 fractions. So it was very satisfying. I won't show it, I have it in a supplementary slide, but we tried to dry these fractions to have the dry fucoxanthine, and we also tried to valorize the rest of the biomass and the rest of the extract with the hydrolysis, etc., to prove that it was possible to finally highlight. How can I say that in English? I don't know. Anyway, 80% of the total biomass of the total extract was valuable. Finally, not only the fucoxanthine. Here, the focus is on the fucoxanthine, but you have many other valuable compounds in it, lipids, etc., that later, after me, they managed to recover. Okay, so after all this solvent system and extraction, the solvent that we changed and optimized, we tried to decrease the quantity of this extraction solvent. So remember, at first, I told you that we extracted 1 gram of dry biomass with 200 grams of solvent, which was, in my opinion, way too much for 1 gram, not viable at industrial scale. So this is what we did. On this graph, you have on the x-axis the different solvent concentrations that we used for to extract 1 gram of dry biomass with the ultrasound-assisted extraction. And on the y-axis, you have the fucoxanthine concentration of the extract in milligram per gram of dry biomass. And by decreasing the quantity of solvent from 200 grams to 9 grams only for 1 gram of dry biomass, we managed to extract 1.5 more fucoxanthine with 20 times less ethanol. Sorry, I left a bit of a French word here in this slide. Anyway, so it was really nice results, especially as I expected to have a very weird paste even before the solvent evaporation. When it is too viscous, it is impossible to manipulate. And finally, with 9 grams of solvent and 1 gram of dry biomass, 1 gram of dry biomass, it's a lot. It doesn't seem, but it's a lot. It was not that viscous, so easily working. I don't know how to say. So I think it could work at more at higher quantities. That's why I meant. So in conclusion of this first case study, so we tried to produce fucoxanthine and develop an eco extraction purification process by using the centrifugal partition chromatography. So first we managed to scale up the cultures of Disochrysis lutea. It's an algae which is really easy to scale up, and it was already done before us, but we just showed in our study that we managed to increase a lot the fucoxanthine content in this in the cells. We managed to replace acetone by the ethanol as an extraction solvent, and we decreased by 20 the specific extraction solvent consumption. which was nice as well. We replaced the methanol of the mobile phase by the ethanol in the centrifugal patient chromatography. And the final solvent system was cyclohexane, ethanol, and water in these proportions 2:1. As I said before, clearly I am not happy with the cyclohexane left in the stationary phase, but in this short time that we had to finish this work, we couldn't try to replace the cyclohexane. Unfortunately, the properties of the cyclohexane are really, really hard to find in a natural and environmental friendly solvent. So a lot of improvement is yet to do for this technique. It's just preliminary results. For us, it was wow, incredible, but still it's preliminary. I am sure that in a few years it will be way improved. And what was nice also, it's that we had only one purification step for 100% of fucoxantina purity. We need to remember that there are lipids. And yeah, I mean, purity among the different, regarding the other pigments. There was no other pigments at all in these fractions. And we obtained finally a very good recovery yield with this technique. So we were quite satisfied with that. So this study is published in Legal Research. Yes, that's it. If you want more detail, I'm happy to discuss them. And especially if you have one day a project about it, I'd be happy to participate because I'm still really interested in it. Okay, so now we move to the other case study. It will be shorter, don't worry, because it's part of my postdoc work. And I've been doing this for a few months. So it's on Pheodactylum tricornutum, which is a diatom. So you can see this pic that I took a few months ago. I was really happy to see the actual tricornutum morphotype. That is really special. It has several morphotypes. So this is a diatom, so a brown algae, bigger than Tizochrysistita. It is from 12 to 14 micrometers. It has no flagella, so it cannot swim. So it's sedimentate if you don't shake it. It's less easy than for Tizochrysistita. And the diatom deposits a frustula. Usually you cultivate the diatom with silicium to allow the algae to build their frustula, but Pheodactylum tricornutum lives very well without silicium, so it's a cost you can save on your culture medium. That is nice to know. And also this algae has a fully sequenced genome, so it's if you want to do some transcriptomic analysis, etc., it's really easy. If you see different forms of cells in your Pheodactylum tricornutum culture, it is completely okay because this algae possesses 4 different morphotypes. The fusiform is the main one. The ovoid is also quite common. The tree radiate, as you have on the photo, it's not that common. And which is what is really rare, it's the round morphotype that I only saw with a highly iron deficiency culture. Pheodactylum tricornutum is also among the best fucoxantine producers. It also produces another omega-3 than Tizochrysistita, which is the EPA, eicosapentaenoic acid, something like that. So it is a microalgae of interest in nutrition, pharmaceutical, and cosmetic, also mainly because of the fucoxantine amount in it. And in this project, so we studied at first 10 ecotypes because there are at least 10 ecotypes in the world, and we cultivate a less common strain that I cannot reveal exactly what it is because this part of the project is confidential. But no problem; it's not a problem for the comprehension of the experiments. So we started with the medium optimization. In a 50 milliliters culture, so very small lab scale experiments. We did a lot of medium optimization and a lot of nutrients, but here we will just focus for this presentation on the iron concentration. The first objective was to optimize the growth and the nutrient enrichment of our medium, which is the common F2 medium. So we tried the different concentrations of iron. But I noticed visual differences in culture color for the same cell density. What is funny is that, so we tested 3 concentrations, the ×1 concentration, which is the common one, divided by 10 times 0.1, and times 100, which I was sure it will be toxic. I was sure about that. I mean, not sure, but almost sure, but finally not. And after 1 week of culture, the 100 times iron culture, they were still white. I mean, it turned into a bacterial culture. I was like, okay. I put them at the back of my incubator. It's okay. I don't do the follow-up anymore, the monitoring, because it's not growing, so it's okay. And I continued with the 2 other concentrations. But after another week, I don't know why, this day, my eyes went on this previous white culture, and suddenly it was completely black. So I completely missed all the process from, it's beginning to grow, to it's completely black. And there was not even a big cell density. That's why it was really interesting. And I told myself, okay, there is something with the pigments here, so I need to do some analysis. So our objective changed. Now we wanted to determine if there was a difference in pigments that was influenced by the iron concentration, which was not described in the literature. Otherwise, I wouldn't have done this experiment, because the first thing I did was to, okay, is there anyone here that already tried these types of concentration? But no. So you can see on the picture the difference of color. I'm not sure we can see really well, but all these cultures are the same cell density. So it's not the visual color. The color, it's not as a function of the cell density, but just the pigments. So this is what we obtained. So we demonstrated that the iron concentrations influenced a lot the pigment production. It boosts actually the pigment production. On these graphs, you can see on the y-axis the content in milligram per gram of dry weight of chlorophyll A, fucoxantina, and beta-carotene. And on the x-axis, you have the biomass. So each experiment was done, each analysis was done at the same cell density, the same biomass, to be comparable, simply. So we noticed that with the 100 times more iron, we had sometimes 4 times more chlorophyll A, 2 times more fucoxantina, and 3 to 4 times more of beta carotene. I'm sure it is the same for almost every other pigments. It's just I just put the 3. The other are not yet analyzed. And what is interesting, as I said, is that it is quite unpublished in diatoms in the literature. It is known in cyanobacteria that the higher iron concentration really boosts the pigment production. So further investigations are ongoing. I cannot tell you more about that for now, because I don't know myself what's going on here. We know that the iron is a big player in the photosystems, but there was no, in my knowledge so far, no direct link between the iron concentration and pigments in the cell. So, but anyway, it is I am passionate about that. I need to do a lot of more investigation to understand all of that. There is only one study that I found where they increased the iron concentration by 5, and they obtained 3 times more fucoxantina in this other diatom, Nietzsche. But on Pheodactylum tricarnotum, usually the people, they study the limitation in nutrition, in nutrient, iron limitation, what are the effects, etc. But we almost never study such a huge supplementation. So these graphs are a bit confidential because this is a publication in preparation. So please don't use them yet. But soon I hope you will see them on Google Scholar or some other sites. So after this iron thing, we did a different thing. So it's a different experiment, okay, just for you to understand. Where now we just wanted to increase the volume for this specific strain, which was quite new for us, and we wanted to harvest the biomass and do a bit of medium recycling, also to respect more of green process experiments. So we scaled up our production in 30 liters culture in a tubular photobioreactor, which was the first try for this strain finally. The first objective was to increase the volume. We keep the normal iron concentration to avoid this latency phase that we had in a 50 milliliters culture. So this photobioreactor was in a greenhouse, so it benefited from natural light plus artificial light. So there was no night, no dark light cycle here neither. And the temperature was, because it was in the greenhouse, and we have a lot of sun here in the south of France. So it varied from 15 to 25 degrees. And we had, of course, a CO2 supply to manage the pH. So as you can see on the picture, the growth was okay. We went up to 4 grams per liter, but it clearly could be more. It's just that for the purpose of the experiment, we stopped it. And this is where it goes very interesting. So the other objectives of these experiments was to have to build a semi-continuous culture by harvesting the biomass and recycling the medium. So the principle of this experiment, of this machine that you have on your right, is that that we concentrated the biomass from 30 liters to 2 liters using a 0.2 micrometers vibrating membrane from the company Sunny Membrane. Maybe you know of it. So the culture is concentrated in what they call the retentate. You can see on the left of the slide, this is the retentate, that is to say the culture at the first, on the far left. And then after a few minutes or few hours of running, you have just a few liters remaining in the retentate. This is the culture, just more concentrated. And on the other hand, you have the clean permeate that is recovered. So you have two, oh, I don't know the word in English, two way out on this membrane, one way out for the retentate and one way out for the clean permeate, which is the clean and cell-free medium that is ready to be reinjected in the PBR for the semi-continuous culture. So we save the water and we save the nutrients that have not been consumed yet. And you can see on the photo that the clean permeate is a bit colored. It is okay. It's because we had some lysis at this time. It is normal, but there is no cells anymore. It's just pigments living their life in this clear medium. It's okay. And so once we recovered the permeate and we concentrated our culture, we centrifuged the liter or the 2 liters remaining, and we lyophilize it to obtain our biomass. And later we can use that for fucoxantina production, for example. This part is not really focused on fucoxantina production, but you can easily imagine what could be done with this technique. So this slide is to show you the semi-continuous culture that we managed to build with this technique. So at first we started a culture in this tubular photobioreactor. It is the same than before. We started at a quite low light and not to stress the cells because the strain is easily stressed by light. We increased the light at day 11, and we can see that immediately the cell density increased a lot. And when we reached 80 million cells per milliliter, we just harvested 10 liters among the 30 liters of the culture. And we did this process with the vibrating membrane. So we obtained 10 liters equivalent of biomass and 10 liters of, 9 liters of permeates that we used to dilute the culture in this photobioreactor. That is the photo that you have here. It is a culture being diluted and being mixing with the pump during the process. And after a short phase of latency, then the cells restart to grow again, and then we harvested again. We stopped here for experimental purposes, but we could have done many different cycles, and I think it would have worked perfectly as a semi-continuous culture. So as I said, each time we saved water, liters and liters of water, and we save the nutrients that are left. And we can analyze, of course, the nutrients to see what's left in the medium, not to enrich too much. We're still in a further industrial objective, so we need to cost save everything. And just to finish, I think, with this case study, this is an example of the biomass concentration within the retentate with this vibrating membrane technique. So it is on the 10-liter harvesting, this one. So it is an exponential curve, of course. And to harvest, 10 liters, it took 1 hour. So it is quite fast compared with other centrifugation techniques, which are really electricity consuming as well. This one is really, the centrifugation part at the end it costs a lot, but it is really short. It's like 10, 15 minutes, whereas this part is really is quite cheap, actually. So I think it's a good way to harvest the culture. So we managed to concentrate 10 to 20 times the culture, and I think it could be more. It's just that we tested with maximum of 30 liters, but and it was a small machine. They have big ones, this company. And the recovery yield of the dry biomass was between 90 to 97%, so very few losses, which is quite good compared with other centrifugation techniques that I used. I didn't tell before, but how we did to recover the Thysochrysis lutea cultures from the 300 liters scobolite. It was with the centrifuge, and it was so long, so long. Okay. In conclusion of these two case studies that I just presented, so first we had this scale-up of fucoxantina production with Thysochrysis lutea cultures. As a reminder, Thysochrysis lutea is a famous algae for the fucoxantina production because it's one of the best producers. We managed to increase the fucoxantina by 6.5. We noticed that the light accessibility was the key factor for the fucoxantina production because, I didn't precise it before. I completely forgot. But the scobolites at the end, you can see these big columns. Finally, the cells in the middle, they don't receive any light. So if you have the same incident light than the 300 milliliters, you don't have at all the same light accessibility for the cells. It's completely different. The bigger your container is, your tube is, the less the light is accessible, which is very good for the fucoxantina production, the photosynthetic pigment production, but not that good for the growth. The growth was really lower in these reactors than the smaller ones. After this production part, the downstream processes for fucoxantina recovery was a set of experiments that allowed us to extract the fucoxantina with a very few amount of solvent, 9 grams of ethanol for 1 gram of biomass, and we managed to have only one purification step to obtain a fraction with 100% pure fucoxantina with the centrifugal partition chromatography. And the perspectives for this, all these experiments are multiple. First, of course, the replacement or at least the decrease of the specific consumption of the cyclohexane, which is really not environmental friendly, or maybe we can just recycle it. It is done for some solvents, so to my knowledge, I think. And of course, if you want pure fucoxanthine, we still need a step to separate the lipids. One green chemistry principle also is to lower the steps of your procedure when you want a product. It's often like 25 steps to obtain the pure molecule. But if you the less you have, the less it costs you, and the less you will use solvents, etc., etc. And of course, we didn't try it. I mean, we did a bit to scale up the process of centrifugal partition chromatography. We used a 1 liter column to test because these results I presented was in 240 milliliters column, so very small. And 1 liter, it was okay. Just a bit more of emulsion that we need to get rid of, so a bit of system instability, but it wasn't that bothering at this time. And then on the case study on the Fiodactium trichonotum, well, we just talked about that, but in a few words, we saw that the iron boosts the pigment production in Fiodactium trichonotum. So we use the F2 medium, with a very high iron concentration compared with the original one, and we managed to have twice more fucoxanthin, 4 times more chlorophyll A and beta carotene. And we wonder, but that it is still to think about, is it direct boost of the iron on the pigments, or is it is the iron boost the other metabolic processes in the cells that themselves increases the metabolic processes on the pigment specifically? I don't know yet, but yeah, I will try to have an answer in a few months now. And about Fiodactylum trichonotum, we managed to build this semi-continuous culture while we are recycling the medium. So we validated the biomass harvesting process with this vibrating membrane that is really practical to use. I don't have actions in this company, I promise. It's just I really like the process. We obtain a really high recovery yield with this method, which is really nice, and we managed to recycle the medium, the water, and other nutrients. Of course, we need to re-enrich the culture sometimes when we dilute it, but that's all. It's only a few. And that's it. Semi-continuous, I already talked about that. And I thank you for your attention. If you have questions, please don't hesitate, even if your English is not perfect. Thank you very much for your attention.